Reproduction is costly in terms of time and energy. In most avian species, costs are involved in establishment of territories, preparation of nests, production and care of eggs, and feeding young. Sex roles and parental investment vary according to reproductive strategy and mating system. In terms of reproductive strategy, avian brood parasites have evolved to reduce their investment in parental care by laying their eggs in the nests of other species, which provide all parental care to parasitic offspring until independence. In terms of mating system, monogamous species are associated with parental care. Both sexes share incubation and feeding young. In contrast, polygynous species, normally males, tend to provide little or no parental care during the breeding season. Costs of parental care are expected to vary with the environmental factors, and one we explore here is urbanization. Urbanization is expanding worldwide at exponential rate and affects avian communities. And whereas it normally leads to increasing population density, the species richness is usually lower than in natural habitat. Urban ecosystems have benefits and costs on the birds' populations. And for example, in urban habitats are characterized to be more constant and predictable in food resources, and also to have less predation pressure and warmer microclimate in winter. However, urban species have to cope with new challenges like permanent human presence, pollution, or high noise levels. Costs of parental care are so high that this part of the life cycle is often referred as being the major stress for birds. Birds respond to unpredictable environmental and social stress by activating the HPA axis and releasing corticosterone in the blood. This is known as stress response. Cork helps protect animals from those noxious stimuli. However, high levels have negative consequences to immune function, cognitive ability, growth, survival, and reproduction. Cork has been usually measured in blood, but however, this method shows some limitations associated with the short period of time, a few minutes, that these high levels are in the blood. Recently, Bortolotti et al. Proved that corticosterone can be measured reliable in feathers. Cork in feathers appears to be stable over time and determines the overall exposure of the birds to the hormone over the period of time, to days to weeks, depends on the species, that the feathers was growing. Our objectives in this study were evaluate the cork in feathers in avian brood parasite versus non-parasite species, evaluate the cork in feathers in polygynous versus monogamous species, and evaluate the cork in feathers in both reproductive behaviors in rural and urban areas. Our predictions were avian brood parasites have reduced their investment in parental care. Therefore, we expect that they should have lower cork than non-parasites. Polygynous males usually incur less cost in parental care. Therefore, we expect that they should have lower cork than their mates and lower than monogamous males. And finally, urbanization leads to environmental changes. Therefore, we would expect variation in cork levels between rural and urban areas. We select 3 sympatric cowbird species: shiny cowbird, screaming, and bailing. Shiny cowbirds are brood parasites, and they are extreme generalists. They parasitize more than 200 species, and one of them is a bailing. They are polygamous, and they are dimorphic in plumage Males are black and females are gray, and in sex, males are heavier than females. Screaming cowbird is also brood parasite, but they are specialists, and the parasite parasitize 2 or 3 species, but almost always they parasitize baby. They are monogamous, and they are more dimorphic in size. Males are heavier than females. And baby is non-parasite, is monogamous, and monomorphic in plumage and in size. In terms of parental investment, in shiny cowbirds, females are who search for the host nest without the help of the males to lay the eggs. And for that, we consider here that the parental investment in males is low. In contrast, screaming cowbird, both sexes go together to search for the host nest. And for that, we consider that the parental investment was medium. And in the last case, baby, both sexes share the parental investment during all the breeding season. And for that, we consider there was a high parental investment. The study area was in Argentina, mate and cow, in 7 localities in the province of La Pampa and Buenos Aires during 2 consecutive breeding seasons in 2006 and 2008. As urban habitat, we were sampling in 3 neighborhoods of the city of Bahía Blanca and Toay. And as rural habitat, we were sampling in 4 locations in La Pampa. There was a semi-arid forest, open forest, and grassland. We used midnet to catch the cowbird. After that, we banded. We took the weight on the wing cord to estimate, to calculate the body mass index. We estimate the age by plumage and the sex by plumage in shiny cowbirds, and the other 2 species we took a blood sample to analyze by molecular analysis. And we collected feather. We collect always the feather in the same position, the pit-tail feather, to analyze the cortical center. We use this new technique to analyze the corticosteroid feathers, and we use Glimmick's model to analyze the association between the corticosteroid feathers and variables like sex, habitat, body condition, and all the interactions. And we control the effect of the year and the location, including these 2 variables, like random variables in the model. This table shows the result from the 3 Glimmick's models was for each species. What we found was that the sex was significant in shiny cowbirds, but not in the other 2 species. The graph shows the corticosteroid feathers in the 3 species. Blue symbols are males and red females. And we can see that in shiny cowbirds, males show lower corticosterone than females and lower than the other 2 monogamous species. And this result would be consistent with the hypothesis that polygamous males with low parental investment, have lower cord than their mates and lower than monogamous males and females. We also found that the non-parasite species show higher corticosterone than the other 2 species, the brood parasite. And those results would be consistent with the hypothesis that brood parasites have lower cord than non-parasitic species because they have lower parental investment.